Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Expanding Access to Gene Therapies Through Novel Cell Lines and dPCR Technologies. I'm Abby Pinchbeck, an editor at BioInsights, and joining me today are Mark Voigt, Jagadish Loganathan, and Andrei Nodshevsky, who will delve into the development and applications of a novel cell line and highlight the pivotal role that dPCR plays as an analytical tool in evaluating the quality and characteristics of AAV. After the presentation, we'll have a live Q&A session, so feel free to pose your questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to these during the session. I'd also like to draw your attention to the Resources tab on the right of your screen, where you can find more information on the topics covered today. Now I'd like to introduce our presenters. Jagadish Loganathan is a highly accomplished scientist responsible for establishing research and development operations at Agathos Fargo, North Dakota facilities. Prior to joining Agathos, he held research positions at Indiana University School of Medicine and North Dakota State University. Andrei Norchevsky is an R&D scientist who leads development and implementation of emerging technologies, analytical methods, and insect cell culture development at Agathos Biologics. He holds a Doctor of Philosophy in Genomics and Bioinformatics from North Dakota State University. Mark Voigt is the current Director of Business Development and Operations at Agathos Biologics. Mark holds a BS from North Dakota State University and an MBA from St. Cloud State University in Minnesota. So without any further ado, I'm going to hand over to Mark to kick us off with today's presentation.
Thank you for that introduction. Good morning, everyone, and thank you for taking time to attend our brief talk this morning. I'd like to take a second to also thank QIAGEN for facilitating our presentation for today and their general overall support. They've been really valuable as a resource for Agathos and addressing any of the questions that we have in our dPCR assay development. And their technical team is truly world-class, so thank you. I'd like to start off our talk today with a little background on Agathos Biologics. It helped give you a better understanding on our approach to expanding access through the development of novel cell lines for biomanufacturing. First, we get a lot of questions on the name, so I just want to briefly mention that Agathos is the Greek word, which means good. It is an inspirational word. We want to both be diligent and do good science, but we also want to make sure that we're focused on our company mission. The idea for Agathos was conceived several years ago, but officially launched in 2021. Our founders are James Brown, Michael Chambers, and John Ballantyne. John and Michael are the founders of Aldevron. They founded that in 1998, and James Brown was a senior level executive at Aldevron and previously he was a second employee at Regenix Bio. Our founders have extensive connections throughout the biotech and gene therapy industry, and they have a remarkable track record of successful entrepreneurship. Some people don't know where Fargo, North Dakota is. We're dead center of the country, way up north. Wonderful area to live. A lot of people don't think of Fargo as a biotech area, but through state initiatives, the biotech industry is rapidly growing within the region. We also, in our community, have 3 universities, so access to emerging scientists is wonderful for the area. Within our facility, we're fully outfitted to work with AAV materials. We offer to customers digital PCR services. We also have a high content imaging device that we offer as a service to customers. So if you're interested in, after this presentation, and learning more about how Agathos can help you evaluate your AAV, please check out our website at agathos.bio. The primary goal of Agathos is to advance genetic medicine and novel platforms and therapeutics and tools. We want to accomplish this by developing better technology and alternative products that avoid any ethical concerns or objections. I like to think our CEO, James Brown, often uses the analogy or draws a parallel with the successful approach of the Impossible Meat Company. Their company was founded by a group of vegans who knew they wouldn't make everybody a vegan, um, but they really offered an alternative, plant-based meat alternative to the marketplace. And through providing a high quality product, they were able to cater to customers for various reasons for wanting an alternative. Um, you know, much like them, we also aim to offer an alternative and options to researchers and therapeutic companies. Our overall goal is to provide new paths in AAV manufacturing that is ethical and respectful of diverse perspectives. We know that we're not going to turn everyone away from existing methods, but we believe there's a market demand and we want to provide a valuable choice or a viable choice for all customers. Here's just a quick snapshot of why we feel there's a market demand. New therapies emerging on the market seem to be in that $2 to $3 million range. We know that there's opportunity for cells that are better at vector production and help reduce cost. With potentially 7,000 conditions fit for gene therapy-based interventions, there's a growing demand for vector. And even though there's tremendous advancements in manufacturing, there's still not going to be enough vector to meet demand. And lastly, we know that people have personal reasons and personal beliefs tied to the manufacturing of biologicals. So we want to make sure that when we manufacture something new and superior to what's on the marketplace, we want to be sensitive to people's beliefs and make sure that we give access to our therapies to all. And lastly, you can't. Lastly, there's a considerable amount of controversy regarding the current cells that are in the marketplace. And again, when we launch our cells, we want to make sure that we're sensitive and avoid all that controversy that is potential. Excuse me. And again, this is a really good slide that captures, what we're out to do. Our goal is to provide alternatives in the marketplace. We want to be able to provide highly effective cells for biomanufacturing. We want to avoid ethical concerns. We're working really hard to produce cells that have superior performance to what's currently available. And we want to make sure that our cells will meet regulatory compliance for gene therapies. I'll now hand it over to Jagadish, and he'll talk a little bit more about some of the cells we're developing at Agathos.
Hello all. Good morning. Thanks, Mark, for giving a wonderful presentation about Agathos and the role of Agathos in future gene therapy. Initially, I thought of presenting the slides to talk about little background or history of AAV and the packaging cell lines, but based on my recent visit to ASGCT meeting for 2023, where we presented 2 posters and listening to various talks and listening webinars through BioInsight websites, I realized that the single stranded virus AAV of 4.7 kilo base pairs branched out to everyone in science and acquired center stage of attraction for human gene therapy. In that case, I realized that it is not necessary to spend much time on the introduction about the AAV and the production. Typically, AAV particles are produced in even complement cell lines, the cell lines transformed by adenoviral even function. For almost 2 decades, human embryonic kidney cell line, that's HEC293, have been the only complementary cell line suitable for production of recombinant AAV, and with a little background about how they made those cell lines. This led to the researchers on developing a new packaging cell lines complemented to even function that will help the researchers to understand how the cell lines are really made, in what way we can improvise the cells for the better yield in the future therapeutics. Here, we introduced a novel cell line, BHK-E1 wild type. Hey, Mark, next slide, please. Thanks. Novel cell line BHK-E1 W2, transformed by adenoviral even function by designing a plasmid in Agathos, and the transformed cell lines are selected by antibiotic pressure hygromycin. So when you look at the slide, The figure A is a representative of how we design the plasmids and how we transfect the cells to make a transformed cell lines. And the figure B represent the expression of E1A from the transformed cells, and the figure C represent how the transformed cell phenotypically changed when compared to the control cells. When you see the microscope image of BHK control cells, you can see the actin filaments running all over the cells. But once the cells have been transformed, you can see the actin filaments have been restricted and the cell number has been increased. So these are the very initial observation of the transformed cells. Mark, can you go to the next slide, please? As everybody know how the AAV is made in HEC293 cells, helper-free virus method by transfecting 3 plasmid at one time and making AAV. Similar approach has been performed here also. We did a triple transfection on the BHK WT1 transformed cells to see whether these cells are able to make recombinant AAV. Next slide, Mark. And we found, and also we presented this data in the ASGCT meeting in 2023. We noted that our BHK-E1 transformed cell lines produced AAV. We first started making recombinant AAV2, and here we show the figures for the capsid and also for the dPCR showing the full capsid. We measured the transgene GFP here. Next slide. And in addition to that, we saw that our cell is making AAV by ELISA and dPCR, and we want to see that whatever the capsid we made is intact. So traditionally, people used to show the three proteins of any AAV material, VP1, VP2, VP3. And we ran a Western blot on those viruses collected, and we saw the 3 individual bands after Western blot for VP1, VP2, VP3. And we also quantified semi-quantitatively for the ratio of the 3 individual bands. And we found that it was almost closely matching to the AAV material made from HEC293. And also to that, one more screening step we need to do that whether the AAV material intact produced is able to infect the HepG2 cells. That's a commonly used technology to make sure the AAV made is transfecting your transgene into their host cells. And we also noticed that our AAV made from BHK cells was infecting effectively the HepG2 cell line. Thereby, we confirm that whatever the AAV made is sure that it is able to transfect the host cells for the transgene. Next slide, Mark. After attending the ASGCT meeting, there are a lot of questions we had. And one of the important questions like that, whether your cell line, the packaging cell line you made is capable enough to make other serotypes commonly used, like AAV2, AAV5, AAV6, and AAV8. And this is the figure we are showing here to show that we made the cell line, what we made can able to make other serotypes of AAV. So when you look at the figure, so you can see ELISA data and the dPCR data, and the third bar graph indicating the percentage of packing. Because one of the hurdles in AAV preparation is that a lot of empty capsids are made. We want to see whether our cell line can able to package efficiently the genetic material for in different AAV. And we measured also based on the AAV and dPCR. We saw that in AAV2, like 15% of packaging. And in the AAV5, we saw 32% of packaging. And in the AAV6, we saw 31.32% of packaging, and AAV8, 17.18 packaging. So these are all some of the initial observations we made with our BHK cell line making different serotypes. So, and the another question is that whether we compare these results with the current cell line. So initial understanding is that we want to develop a new cell line. In some of the ways, our cell line is not performing better with the HEC293, but it is the alternative cell line. It needs to do a lot of work on improving this cell line. So we take this opportunity. Hey, Mark, next slide. Next slide, please. So we take this opportunity, the presentation, to show that we have a new packaging cell line. It needs to be done a lot of work. So thereby we continue to develop the cell line for a better yield and better performance. And also we are ready to share this cell line with the collaborators. So thereby we can have a better cell line in future apart from HEC293. So I'm just done with the presentation. Thanks, Mark. I hand over to Andre. So he will talk about the new cell line and the development of new technology for analysis.
Hello, everyone. Thank you for joining us. Thank you, Mark and Jagadesh, for your introduction. Here, I would like to talk about dPCR more from the perspective of the research and development scientists and assay development to describe our newly developed BHK1 line. Next slide, please, Mark. So for general introduction about dPCR and what lies behind is that new technology developed by QIAGEN. QIAcuity device uses so-called nanoplates, which is populated with nanopartitions. There's thousands of nanopartitions on each plate, and each nanopartition idea is to basically distribute DNA of your sample to those nanopartitions and where PCR amplification will take place. This nanopartitions where in case where there is your target will have a positive signal, amplification will take place. Partitions without the target will be so-called negative partitions, and then you can observe differences between, then you can calculate the number of positive partitions for your sample and then calculate copies per microliter. One other advantage of the dPCR is the use is so-called absolute DNA quantification. In this case, you do not have to provide and prepare a standard DNA standard to quantify your samples. Next slide, please. Why we chose QIAGEN QIAcuity dPCR? In general, it's a very competitive device as far as the pricing, and as Mark mentioned before, we have very good experience with the QIAGEN customer service, especially in the early stages of assay development for AAV quantification. This device allows us to use up to 5 fluorescent dyes for development of our various assays. It's relatively fast to provide our results. Up to about 2 hours, we get our samples quantified. And it allows quantification of a wide range of the samples, from 24 samples at a time to about 1,248 samples, depending on the device used. And QIAGEN provides devices which can assess 1 plate at a time, 4 plates, or 8 plates at a time. Next slide, please. So in general, in development of the BHK1, we assess AAV titer at the various stages of the process development. In this case, just directly after AAV production, we assess crude lysate samples. Those samples were later purified by affinity chromatography, so the purified product was assessed. In later stages, we are going to assess titer of the AAV after ion exchange chromatography. Those numbers, those assessment of those samples in conjunction, in the connection with the ELISA, allows us to assess full empty ratio. At this point, we are using simplex assays for the EGFP, EZ40, and other single targets. But however, we are moving toward using the multiplex dPCR to assess genome integrity of our produced AAV. Next slide, please. As I mentioned, I would like to present our experience with dPCR from those early stages of the assay development, or the early adapter of the dPCR technology. Here, there's an experiment where we demonstrate amplification or a percent recovery of the ATCC standard. So in general, when we purchased our dPCR device and develop quantification assays, we wanted to see how those assays perform. So we used the ATCC standard, AV2 standard, to assess a percent of recovery of that standard. In the first column, you can see PBS diluent did not produce any, and this is NTC, so there's no template control. Second was AAV standard spike with the G-block. G-blocks contain the same targets as the AAV genome and no DNase treatment. Second, RSS standard with G-blocks with DNase treatment, and then 3 technical replicates of the standard itself and the mean. What you can see that non-DNase treatment of standard and G-blocks produce as expected numbers. In the third case, the same sample treated with DNase provide only the titer expected from the genomes of AAV encapsulated. However, as you can see, the recovery of that standard was about 56% comparing the C of A stated titer. When we search literature and related titers obtained from dPCR or the dPCR, and we applied the correction factor, basically, you can see how those technologies differ as far as amplification of that standard. We obtain about 84 and 96% of recovery of that standard. Most likely, the differences between titer of the original titer stated at the ATCC standard, AAV standard, and the new dPCR and dTPCR is due to the old qPCR method used to quantify that standard. Next slide, please, Mark. And also, as far as the nitty-gritty details of the method development, here I would like to present 2 slides where I literally took 15 independent dPCR run that happened basically from a few weeks ago to basically a few months earlier. As you can see, those, again, 15 independent runs by 2 independent analysts performed with multiple technical replicates. And overall, AAV2 standard use in this case showed 84% expected recovery, plus minus 12% standard deviation. However, what I would like to point here that technical replicates for dPCR are very, very highly reproducible, with occasional, I would say, under and overestimation. The most variation, actually, what you can see here, happened at that orange box on the right side, when we actually introduced to this method new analyst, and this is actually literally the training sets were included here in that box. So yes, there is a variation. However, as I pointed, this is the new person setting up that reaction, and overall, I was very happy. Even if there was a variation, it was not very significant. Next slide, please. So this is the same set of independent dPCR runs. However, in this case, we use the SV40 poly-A region of the same AAV control. Again, situation similar as before. However, in this case, you observe 100%, literally 100% of the percent of recovery of that standard, plus minus 14 deviation of that. It's relatively common to see in a qPCR in general that the same template amplified with 2 different sets of primary probes assay will have a slightly different percent of recovery. Could you skip to the next slide, please? Here, I would like to briefly discuss results of the collaborative work Agathos did with the QIAGEN on the CGT viral vector lysis kit. This kit was developed to help with the AAV capsid lysis. And we tested that kit with 2 insect cells, SF9 and TNS42, and 2 mammalian lines, HEC293 and BHK21. And also, we used that kit to lyse AAV2 diluted in a dilution buffer. And as you can see, AAV2 standard used for that spiking experiments did not show differences between different matrices used to evaluate AAV genome. Could you please skip the next slide? Similar situation was observed for the AAV5 standard spike to those 4 different matrices. Only plus minus 10% of the standard deviation from the percent of recovery of the standard was observed. This indicates that dPCR viral vector lysis kit allows proper capsid lysis and is not very sensitive to different matrices that AAV was evaluated. And overall, we found that dPCR is very robust device to evaluate AAV in a lysed cell lysis, purified samples, and we are going to develop, like I mentioned before, assays which will be targeting multiple fragments of the viral genome to test AAV genome integrity. Could you please, next slide, please? And here I would like to especially say thank you to Pavle Matak and Daniel Lovgren and Dominika. They were essential in helping us in development and confirmation of our AAV titration assay, and in particular in a work related to the assessment of their capsid lysis buffer. And thank you very much, and we are open to questions.
Great, thank you so much, Mark, Jagadish, and Andre, for a great presentation. We're now going to start our Q&A session, so if you'd like to join me back on camera. And Mark, if you stop sharing your screen there, wonderful. Just a note to our audience to please keep your questions coming in, and we'll get to as many as we can and answer any others by email. We've got some great ones in already, so I'll just launch straight into the first one. Because they are of non-human origin, can you discuss any possible regulatory hurdles that arise from using a BHK line in production? Jagadish, I believe this one's for you.
Yeah, it's a good question. This is the recent question. I also read about that one. Yeah, BHK is a non-human cell line, but it's already two proteins have been approved by FDA for hemophilia disease. So it won't be a much problem, and lot of vaccines have been produced using a BHK cell line. So it won't be a much hurdle in approving the AAV for in future.
Thanks, that's good to know. And how do your BHK cells respond to AAV serotypes with more exotic and heavily engineered capsid constructs?
Yeah, those studies need to be addressed. So far we are currently working with a screening process. I don't have an answer at this time. Maybe we need to do a couple of more experiments to address those questions.
No problem there. And Andre, I believe this one's for you. What conditions do you test when moving an assay from qPCR to dPCR? Do you have any tips for optimization?
So in general, when you move between qPCR and dPCR, primers or probes or in other way assays should perform similarly as far as the temperature or annealing temperature, since all those methods relies on the PCR amplification itself. So in this case, I would say dPCR optimization will be similar to qPCR or even PCR reactions.
Brilliant. And why do different AAV backbone targets produce different readouts?
So it kind of relates to the first question you just asked. So in general, PCR action relies on binding of the primers and probes to the DNA template. And in general, I would say different ways or TMs or TAs of the primer and probe will affect slightly that amplification of that targets between those two different regions of the DNA. So it is not uncommon to see that the same template, the same piece of DNA, when probes and primers are designed, even if close proximity, will produce slightly different amplification levels. So this can be elevated by optimization and redesign of the probes and primers. However, I would say those 10%, 15% differences are something to be expected. And of course, this could be improved, but yeah, it's, in my opinion, personally, very minor difference.
Great. And I think we've got time for just one final question here. Can you please discuss any possible reasons why the dPCR copy number is different from the stated ATCC COA?
Could you please repeat the question again?
Can you please discuss the possible reasons why the dPCR copy number is different from the stated ATCC COA?
Oh, yes. So in general, ATCC standard is one of the oldest available AAV standards. And it was, as far as we know and learn how the quantification was performed, first of all, it's a qPCR, but also we noticed that protocol for the qPCR was relatively old and not very optimized for AAV. So if you think about it, AAVs AAV genome is encapsulated in protein. So there is a multiple steps you have to perform first when you start assessing the genome. So in this case, you have to have a DNA treatment to digest any DNA that was released released accidentally, in a sense, from the capsids. Then you have to do the capsid lysis, and then you have to prepare the PCR reaction or qPCR reaction to the dilutions. So this is multi-step process, and many things can happen improperly, and the tighter differences between, let's say, brand new, newly developed dPCR or even the dPCR method can be slightly different than this for qPCR. So then, in general, that'd be the simplest way to say it's the differences between the protocol preparation of the AAV.
Great. Thank you so much for both of you for answering those questions. That's unfortunately all we've got time for today, but any questions that we didn't manage to get to, we will reply via email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. All that's left is to thank Mark, Jagadish and Andre once more for a great presentation today, and thank you to our audience for listening. We hope you'll join us again soon.
Thank you.
Thank you.